So we'll start in 5 minutes. First, let's join all the students and we'll start. So hello everyone. Myself Ankit Malik from IIT BHU. Happy हमारा fellow. So let's start this open session for all. Still students are joining. So does anyone have any doubt from the last live session or from last assignment? So they can ask one by one in chat box. Then we'll start one presentation on DNA nanotechnology. Meanwhile, you can drop your questions in chat box if someone has any. Or let me start the presentation then. Does anyone have any question from the last, from nanoparticle synthesis characterization? Okay, so it's no. So let me share my screen, then we'll start the presentation. So my screen is visible to all. Okay, so let me start. So today we'll discuss about DNA nanotechnology. My screen is visible to all now. Confirm.
Yes, it is visible.
So let's start. So DNA nanotechnology. So welcome to the world of DNA nanotechnology. So here we explore the fascinating potential of DNA molecules as building blocks for advanced structures and devices. So DNA nanotechnology is also known as nucleic acid nanotechnology because DNA contains nucleic acids. So it is a fusion of nanotechnology plus biotechnology. The combination of nano and bio leads to DNA nanotechnology. So design and fabrication of artificial nucleic acid structures for technical applications in DNA nanotechnology. So DNA is the carrier for genetic information, but here used as a structural component for the synthesis of nanomaterials or nanostructures. So let's discuss about DNA. What DNA exactly is? So DNA is a polymer of deoxyribonucleotides. So there are two types: nucleotides and nucleosides. Combination of sugar, base, and phosphate is called as nucleotide, and sugar and base is called nucleoside. So DNA is found in chromosomes, mitochondria, and chloroplasts. So it's, as you can see, the phosphates, sugar, and base. This three will lead to nucleotide, and this sugar and base is called nucleoside. So DNA and RNA both have 5 carbon sugar, known as pentoses. DNA contains 2-deoxy D-ribose, and RNA only contains D-ribose. And there are two terms: pyrimidines and purines. So cytosine, thymine, and uracil is known as pyrimidine, and adenine and guanine is known as purines. So this thymine is only used in DNA. As it comes to RNA, it goes to uracil. Some students are waiting in lobby. Let me edit. Hmm. So why DNA? Why DNA is used for nanotechnology? Why DNA only? Like it is very stable, relatively stable chemicals than other chemicals, which exist in different forms such as nucleotides, nucleic acid, as we discussed earlier. So as a polymer, it can form very long molecules. It has a well-defined and repetitive structures of DNA, nucleotides and nucleosides. So rules for determining the structures are very simple and well understood for DNA. In case of DNA, so within the molecule, many atoms are available to form useful interactions or modifications. So DNA is a biocompatible material. So that is why we are using DNA as a structural material for developing various nanostructures. So now we'll discuss this in brief. So DNA has an information storage capacity. DNA can store a vast amounts of information in a compact form. Its four-letter code ATGC can encode digital data, making its potential candidate for high density data storage. That's why it can contain a lot of data. It can use for like a memory card type of things. That's why DNA is mostly preferable thing for nanomaterials or nanostructures. Self-assembly. Self-assembly is the most versatile thing in this DNA. So DNA molecules have a natural tendency to self-assemble into specific structures, driven by the complementary base pairing between nucleotides. Like A will only attach to T and C will only always attach to guanine. So this is known as Sharjaf rule, that A will always attach to T and C will always attach to D. So this property allows scientists to design and program DNA to fold into precise shapes and structures, which is a crucial for nanoscale construction. So precision in DNA technology allows for a nanoscale precision. Researchers can design DNA sequences to create specific structures and patterns by using this precision accuracy data, enabling the constructions of custom-made nanoscale objects and devices with high accuracy. I think some students are waiting. So moving ahead. So biocompatibility is also one of the most uh, relatively factor to this by use DNA in nanotechnology. So biocompatibility. DNA is a biocompatible material, meaning it is generally well tolerated by biological systems. This property makes it suitable for use in medical applications such as drug delivery system or targeted therapies. So in targeted therapies, it will only go and attach or target the targeted site. So that's why it is well used in nanotechnology. Then functionalization. DNA can be modified with various functional groups and molecules, enabling the attachment of other nanoparticles, molecules, or sensors. This functionalization expands the range of applications for DNA-based nanotechnology. Then other factor is selective binding. DNA molecules can be engineered to recognize and bind to specific target molecules with high selectivity, making them useful for biosensor diagnostics and molecular recognition application. Scalability. DNA-based nanotechnology is highly scalable, allowing for the production of a large number of identical nanostructures with relatively low cost and efforts. Then biomimicry. DNA-based structures can mimic biological processes and structures, for example, DNA origami. I'll tell this in detail about DNA origami. So DNA origami can be used to create nanoscale containers or carriers for drug delivery, mirroring some aspects of natural viruses or cellular structures. So this all are the factors that why we use DNA in nanotechnology. Let me see if someone is waiting or not. So DNA nanotechnology is a field that utilizes the unique properties of DNA molecules to construct nanostructures with precision and control. DNA used as a structural material rather than as a carrier of biological information to make structures such as DNA origami methods. DNA origami is a technique that uses a long single-stranded DNA scaffold and short staple stranded to fold DNA into predetermined shapes. Basically, in DNA origami, it can shape like anything. I'll explain it further. Then this method enables the creation of 2D and 3D structures with remarkable precision. So this is DNA origami. Likewise, we use paper like to modify it in different various structures. Likewise, DNA is used to construct in different structures. So here we can use star-shaped, circular, rectangular, triangle. So the first one is the upper one is the images on top are the computer produced folding sequences of the DNA standard, and the bottom row is the atomic force microscope image of different DNA origami shapes. The first one is the shapes are made from computer, and second one is the images from taken from atomic force microscopy. AFM, which I have told in the last meeting, this is the same AFM. So in the B section, it has been shown that the different shapes can elect different biological effects, The triangular shaped origami provided the best accumulation. This is an example. Like this triangular DNA origami is used for DNA for breast cancer treatment. So here the breast tumor site is compared to the square and tube shaped origami. So here in breast cancer case, the triangular DNA origami is best preferably used in as with respect to tri- circular or this star shaped or rectangular, whatever you can say. So this triangular DNA origami is the best preferably used for breast cancer. So this is the example as you can see in 3 in the B section. Hmm. So moving forward, DNA nanostructures. Basic geometry and thermodynamic properties of DNA are well known and predictable by available softwares like TileSoft and GenoCAD. So we can predict about this thermodynamic and DNA geometry by using this softwares available in the market called TileSoft and GenoCAD. So DNA ladders like structures provides the key framework to the scientists. It is existence of commercially available modifying enzymes. Persistent length of DNA is about 500 Armstrong. Then self-assembly property of DNA is easy to construct the variety of structures in predictable manners. So assembled structures can be characterized by various techniques like AFM, TEM and cryo electron microscopy. So this AFM one I've shown the structures now in this one. So this is the AFM microscopy structures. Likewise, we can characterize this by using cryo electron microscope or transmission electron microscope. So DNA nanotechnology. This is I have divided this in two parts, like whatever used in self-assembled DNA structures and in inorganic nanomaterials. So in inorganic nanomaterials, we only use this gold material, two-dimensional nanosheets, fluorescent nanomaterials, magnetic nanoparticles. But in self-assembled DNA structures, we use DNA origami, DNA hydrogels, signal amplification, and deoxyribozymes. Now we'll discuss about the applications of DNA nanotechnology. DNA is the best nanowire in existence because it can self-assemble, self-replicate, and it can adapt various states and conformations. So rapid disease detection using the DNA nanotubes and molecular treatment is very easy by using this DNA nanotechnology. So DNA robotics, wide range of uses ranging from building block robotics. So DNA nanochips, it is used in making such smaller microchips than the current semiconductor fabrication technology. Then DNA computers. Nowadays, this nano DNA computer is there. Maya 2. When you Google this Maya 2 now, it is made by DNA semiconductors instead of the silicon based semiconductors. So DNA can be used to make computers instead of silicon based on DNA stem loop controllers. So these DNA stem loop controllers are used for making these computers. Replacing the normal silicon based circuits, this chip has DNA strands to form the circuit. The example I told that Maya 2 computers. And DNA walker. DNA nanotechnology can be used to create walking bipedal nanorobots. So DNA nanotechnology represents a groundbreaking approach to nanoscale engineering. So its ability to create custom design structures and functional devices has the potential to revolutionize various industries. So this much. So now if anybody has any question, they can drop the question in chat box. Then we'll discuss on that. Also, if someone has any doubt from the last meeting, they can also ask. Yes, Kinjal, I can share this presentation. I'll share this presentation after this meeting. I think I have the email IDs of all the students who have joined in the last meeting. So I'll share this presentation. Shruti shortly believe Shruti you have joined by 7:05 lately. After that, students will ask questions and it will automatically get revised. Don't worry. So I was unable to attend the previous meeting. Is it possible to share both the PPTs? Okay, Vidya, I'll share both the PPTs with you. Actually, last meeting was about the synthesis of nanomaterials, its characterization, and its data processing. So in last meeting, we have covered all the characterization instruments like UV, nanos, zeta sizer, zeta potential, AFM, atomic force microscopy, scanning electron microscope, transmission electron microscope, Raman spectrum. So this all the topics were covered in the last meeting. So still, if you have any doubts regarding that characterization material, characterization things, characterization instrument, you can ask. Hmm, Sejal Persana. So I'm sorry to move my question, but I have a question regarding the registering mail for exam. I already paid the fees and now I'm not sure about that. So Sejal, you just do one thing. You just mail to the NPTEL coordinator of this course or to the NPTEL directly. So they will revert you back whatever it is possible. Shubhajit Pal, sir, how DNA origami works in controlling breast cancer? Could you explain it to my cousin Sean? Yes, Shubhajit. Wait. So here this DNA origami is used to treat breast cancer. So here, as you can see, firstly they have prepared or developed this triangle DNA by using the softwares. Then they have conjugated this DNA origami with this drug DOX. This is called as doxorubicin. Doxorubicin is an anti-cancer drug. So they have conjugated this DNA origami with this doxorubicin, which increased its biocompatibility or its efficacy. Then it is partially digested doxorubicin in DNA origami. So by this mechanism, they have treated it is targeted to breast cancer things. So that's in this way they have treated this breast tumor using this DNA origami. Still, some students are joining. Session recording can you get actually I'm not allowed to share this recording session to all guys so that so those who want the this ppt or the previous videos can they can drop their email ids in the chat box. I think I do not, maybe I have the email ids or maybe I don't know, I'm not sure about that email id. They can drop their email ids in chat box and I'll personally mail them all the ppts and all the presentations. Ravan. Yes, Ravan. Just mute your mic and ask your question in chat box. Okay. Shruti, you can ask question from the last meeting also that I've told now whatever the topic I have covered is characterization instruments and its sample preparation and data processing. You can ask if you have any doubt. Was not present in the previous. Yes, Nitu Sharma. I have some doubt with also last presentation. Okay, I'll share the last presentation also. So let me announce once again. So just drop your email IDs all those friends whoever want this presentation or the last presentation. I'll share the presentation this to this email IDs whatever they are dropping in the chat box. Meanwhile, if someone has any question, they can ask. So those who have joined late, I think I should revise this. Actually, half of the students joined after the conclusion of this meeting. Let me shortly revise this presentation once again to all the students who have joined late. Still, some students are joining. So let me revise once again. So in this presentation, we have covered the use of DNA in nanotechnology. So this DNA nanotechnology is also called as nucleic acid nanotechnology. Basically, it's a combination of fusion of nanotechnology and biotechnology. So you this is some basic information of DNAs. It's nucleotide, nucleosides, and its pyrimidines and purines. So then we have covered the reason why we have used DNA only in this nanotechnology part. So there are some various factors by which are wait, hmm. So these are some factors by which DNA is used for nanotechnology, that is information storage, self-assembly, precision, biocompatibility, functionalization, selective binding, scalability, biomimicry. Then this DNA origami method is used. This is some information about this DNA origami. The images on the top are the computer produced folding sequence of the DNA strand, and the bottom row is an atomic force microscope images. And the second one, the triangular shaped origami provided the best accumulation at the breast cancer site as compared to the square tube and shaped origami. So these are the nanostructures softwares by which we can this biogeometry and this thermodynamic property of this DNA can be targeted out. Styles of in GenoCAD. Then we have covered some fact this self-assembled DNA structures, this DNA origami, DNA hydrogels, signal amplification, and deoxyribozymes. We have covered some application of DNA nanotechnology, which is self-assemble, self-replicate. So then DNA nanotechnology is used in DNA robotics, DNA nanochips, DNA computers called Maya 2, DNA walkers, and etc. So I think everyone has dropped the email ID in the chat box. Can we use this technology in other than cancer diseases? Yeah, exactly. We can use by developing DNA origamis. We can use this DNA nanotechnology in different diseases also. Sir, I was unable to complete the assignment. One due to semester exams. The exam related and assignment related queries you can directly ask to the NPTEL coordinator or this course coordinator directly by email, by texting them mail. This session is for doubt session related to your course only. No explanation for your assignments and exams will be given. So, doxorubicin DNA origami, how is it able to target breast cancer and importance of partial dilatation of doxorubicin DNA origami? Okay, so let me open the DNA origami part. So here the first they have developed this triangle DNA origami. Then the doxorubicin intercalation is done. Then doxorubicin is conjugated with the DNA origami of the triangle DNA origami. Then some partial digestion of doxorubicin DNA has been done. So I'll share this complete article with you. You can drop your email ID also. Vanita Surinder. So anyone has any other doubt? Okay, Vanita, I'll share the article, the related article about this. Still some students are joining. Can you explain the role of biomedical engineer in DNA origami? See, DNA origami is a engineering process. Wait, let me explain. So this the shapes, different shapes of DNA origami is a biomedical engineering part only. So based on the requirement, based on the disease, based on the treatment, the biomedical things are engineered by to design this DNA origami. Kansha. Some of the students has raised their hand. Ashi Pal. Ashi, do you have any questions? Final grades of so Shangmugham. Are you posting that assignment questions in the chat box? So are these the same question asked in the assignments? The DNA nanostructures will have some same function as DNA. If yes, then how will it bind to cell? Okay. It will not directly go and bind or attach or conjugate to the cell. I told you DNA origami, the mediator DNA origami will prepare, develop, then it is conjugated with different drugs, different APIs, active pharmaceutical ingredients, or active metals, like silver, gold, this kind of thing. Then only that DNA will go and attach with the targeted bind, target binding sites. Directly, DNA will not go and attach to any of the cells or any of the targeting binding sites. It requires a mediator. So DNA origami is a mediator here. Cells will not recognize it as a foreign. Cells will not. That's why it's the research now. So different shapes. That's why it's like if we'll try the square and the star shape, then circular, then rectangular. Different types of different structures of DNA RNAs will be prepared and will be targeted. Then it will be checked if it is working or not. If it is providing the effect, if it is efficacy is not, or if it is effective or not on that disease or any tumor, whatever the disease it is. If I want to control the DNA and structures from outside of the body, is it? No, it is not possible. Arunanthi. So, yeah, you are asking like is it a remote control thing? So I'll share this presentation with you all the guys. And how many students are there? Ashipal, uh, she raised. You raised your hand now. If you have any questions, you can ask. Ashipal, you raised your hand. So I think there's no questions further. So let's conclude this meeting. So you can leave one by one. So thank you guys. Thank you so much. You can leave now. Sir, can I ask one personal doubt? Ask Karun, what is your doubt? Vinita, I'll share that article with you after this meeting. If I want to do a mini project based on this course, what are your suggestions for that? Mini project based. You can go for it. First, you have to look for a supervisor or any professor who is working in the same project on the same manner or same profession. So then you can ask them. Then you can start a mini project also. You can ask first. According to me, firstly you have to write one review article related to this DNA nanotechnology topics. Read some articles, read some research articles, review articles, and firstly go with the review article. Then ask one professor, approach one professor. Then you can start your work in the same manner, on the same profession, on the same things. Anybody else have any questions? You can also ask. Okay, let me conclude this meeting then.